So good afternoon. I am not a geneticist, but I'm going to attempt to talk about some genetics. So it's good to be back after a several-year absence from the CSF Symposium and the Think Tank, and it's just great to see the work that's been going on out there. We've been collaborating with Gabe and his group and Dave for a while, and we've been asked to sort of share some of our findings and our results, and hopefully arrive at a way to figure out how we get from a double helix to a significant Chiari malformation. How do we bridge that gap? What's the strategy? How are we going to do this? So I owe a great deal of debt to my collaborators at the University of Utah, and the top two, Lisa Cannon Albright and Craig Tierlink, work at the Utah Population Data Bank, which we'll talk about, which is a unique resource that we have in Salt Lake. And then Deb Nicholson both works with the UPDB and also with the Utah Genome Project, which brings its own resources to bear on this problem, and at the Huntsman Cancer Institute. So we'll talk about the problem, some of our unique resources, what's the evidence of genetic contribution, sort of an approach that we're considering going toward, and then potential study designs. And really the problem is that searching for the human genome, or searching the human genome for genetic causes of disease, like looking for one misspelled word in all of the books in the Library of Congress. I mean, that's what we're after. And it's literally a needle in a haystack. And there's multiple reasons for this. And we tend to focus on the DNA, but we know there's coding versus non-coding regions in the DNA. They can have control on each other, different RNA expression levels and how the proteins are expressed. Are there biomarkers in the proteomics of this problem? Are we looking at a musculoskeletal set of genes, developmental genes that are causing this issue? Is it neurodevelopmental? Is it CSF hydrodynamics? I don't think we really understand this. And if we're going to look at a tissue assay, should we be obtaining blood, some sort of mesenchymal tissue, dura, or tonsillar tissue, neural tissue? What's the assay here? And in my personal opinion, I think this is, at least the complex patients that I tend to see, it's probably some sort of developmental regulatory protein that gets turned on or turned off, whichever way it goes, early during embryogenesis, and then it is switched off the other direction, and the patient goes on and lives his life. So this is incredibly complex, but this is what makes it really fun and really interesting. So our unique resources at the University of Utah, the Utah Population Database, it's a genealogy that was started in the 1970s, and as you know, the LDS population in Salt Lake City is famous for defining their genealogies way back to the 1850s in this case. So that's where this UPDB genealogy starts from. At this point, there's 2.5 million individuals with at least 3 generations. And the most important thing is that this data is linked to the medical data for 2 of the largest healthcare providers in Utah, the University of Utah and Intermountain Healthcare. And this is, and for patient privacy issues, this is like getting into Fort Knox. I mean, getting this data is extremely difficult, which makes these studies challenging, but it is obtainable. And we said, okay, well, Chiari malformation, great, sounds interesting. So we gave them the CPT code, everybody, 61443, I don't know, Jerry knows it, but anyway, CPT code, yeah. And the ICD-9 code for Chiari malformation, and we searched it. And we came up with 2,500 Chiari cases since 1994 with genealogy and an MRI confirmation. They sampled this, and we took 200 of those patients. I personally went through them, and it turns out that only about 80% of those were true Chiari cases. And the reason is that they look at radiology reports. So if the radiologist said no evidence of Chiari malformation, that counted as a Chiari malformation. Or there was a Chiari 2 malformation, and that was counted too. So it's a little bit of a dirty data set, but it's the best that we have. And so if you take it, that's about 80% of these cases are really true Chiari cases. That's what we have. So here's a typical pedigree that this would be a high-risk pedigree from the UPDB with the cases shaded below, but this is typically what we see. And then we looked at this with our group at the UPDB, and we had this published in 2016 in Journal of Neurosurgery, the population-based approach. And this is the meat of it here. And so we identified a little over 2,800 cases with genealogy at 2 major healthcare centers. So the estimated relative risk for the Chiari-1 in versus normal controls. So really, for first-degree relatives with this number of patients, observed versus expected, it's highly statistically significant. So this would be first-degree blood relatives. Second-degree relatives, not so much, but a lot of those were grandparent to grandchild, so it has less of an effect. But with third-degree relatives, which is more important in this circumstance, because you get cousin pairs in this circumstance, it's again highly statistically significant. So you look at this, like Gabe said, is it genetic or is it not? Well, it probably is to some degree. In a large population database, there's definitely a genetic basis to Chiari. So when you look at it in a different way, you see on the red, it's 1,000 sets of matched control cases in our 2,871 cases of Chiari. You see these big spikes, and here's the pairwise genetic distance of first, second, third degree relatives all the way down. So at first and third degree relatives, you see these spikes of increased incidence. And so we are really focused on these high-risk pedigrees, and they're out there. And in this population data bank, there were almost 300 of them, and this is highly statistically significant also, anywhere between 2 and 79 cases. These are all non-syndromic. I mean, we're assuming they're non-syndromic. There might have been some of them, but I mean, it was unlikely given our search methods. And here's a typical case.
If there were open circles or squares, were they MRI'd, or do you have good, I mean, can you say they were unaffected, or you just...
Again, it's linked to the health records, so I don't know if those patients had MRIs or not. There was a radiology report or a mention of it within the health record. So
nothing is going down a family line there. You don't have, it's just heredity news that goes from one generation to the next. Unless you have the information that, I mean, the open circles, if they're unaffected, you know they're unaffected, then you could say, well, it didn't go. But if you don't know, then the other people might have.
yeah, it's possible. I mean, you could have some of those open circles could be affected individuals for sure, but those are the ones with closed circles or squares are affected individuals by the health record. I mean, that's the limit of the search, right? I mean, that's the methodology. So there's 3 critical elements to this type of approach, and to create it creates a very powerful and innovative way of doing this. And you're able to discover statistically validated extended high-risk Chiari-1 malformation pedigree resources, and you're really able to look at candidate variants, and you're able to segregate them out using these high-risk pedigrees. And that's part of the process here. So our approach that we're going to take is we're going to do the same search, but we're going to confirm all of the cases, all the 2,000 cases that we have. They're going to confirm by MRI. We're going to talk about how we're going to stratify those by radiographic phenotype. There's really no ascertainment or referral bias because the entire Utah population is included. And each pedigree has statistically significant excess of Chiari-1 malformation cases. And most of these pedigrees have at least 6 cases, and the families go at least 4 generations deep. So here's our study design, here's our aims. I'm going to go through this fairly quickly. We're going to sample the entire population, and we're going to do, the goal is to bank these individuals, bank their blood or their tissue, and we're going to do whole genome sequencing for 50 Chiari-1 malformation affected cousin pairs from high-risk pedigrees, and use a unique and powerful study design to identify these predisposition genes. So it turns out if you sequence somebody, they'll have potentially 300,000 variants. If you take two cousin pairs, or two cousins that are paired, they share 12% of their DNA. If you're able to match those two patients, you cut that 300,000 number to approximately 5,000, which is a more manageable number to look at and manage statistically. So we're going to test the candidates. It's hard to read here. In 1,500 additional Chiari malformation cases and 1,000 controls from other data banks, such as the ones at WashU and in Spain. So the roadmap at this point is to agree upon and identify certain phenotypes. And these are things I've been talking about with other individuals. And in my mind, there's probably potentially 5 different Chiari phenotypes. And one is just the classic Chiari malformation, which is the tonsillar descent. The intermediate phenotype includes the 1.5 malformation. Gabe just presented evidence from the WashU group that this may describe a different population. And then the complex phenotype is something that we've looked at, and it has the obex position, which is low, and then some sort of skull-base abnormality. So occipitalization of the atlas, a retroflexed odontoid, cranial cervical kyphosis. We're going to separate out the connective tissue disorder-related population, and any mention of a syndrome, we're going to separate that out as well and try to get as clean a phenotype as we possibly can. So we're going to confirm and sort the phenotypes, and we're going to perform whole genome sequencing on these cousin pairs and high-risk pedigrees. We're specifically going to focus on the intermediate high-risk phenotypes, especially with young patients, and then perform the bioinformatics analysis, try to get down to those 5,000, and then test the candidates against these other cases in our population in other centers as well. So at this point, we have 150, every patient who walks in with a Chiari to our center is invited to enroll in this study, both the UPDB study and the Utah Genome Project study. We have 150, this was of January of 2018, 150 families enrolled with 148 probands. You can see there's several trios in there. It's hard to know the exact number because we have different mothers and fathers. So I would estimate probably 60 to 70 trios, and other siblings have been included as well. Our other partners besides WashU is Vanderbilt, Stanford, and Nationwide Children. And we've really tried to form this group called the Chiari Genetics Research Group. And we've invited centers that have the capacity to approach patients, obtain DNA, store the samples, and have them go on to be studied with the overall group. So this approach that the UPDB has taken over time has yielded significant results. And here are some of the examples of the BRAC1 and BRAC2 genes and the GOLM1 gene for melanoma and the RAB10 gene for Alzheimer's. This is the exact approach that led to this gene discovery in these certain diseases. So we're optimistic. We're not 100% sure where this is going to take us, but we're excited about the journey. So I'm happy to answer any questions. John.
It's exciting, but it's like with breast cancer, we don't demand that breast cancer have one mutation. You might have 5 or 10% of the people who have genetic breast cancer. It might be like that for Chiari 1 too. You might just have 5 or 10%, and the rest are sporadic, so that makes it even more difficult.
Well, I think in our mind, it's the phenotypic definition, the radiographic phenotypes are going to be critical to this, really getting a clean phenotype and deciding on that, and just, okay, are the genes segregating out into different phenotypes? And I think that's going to be the most powerful thing we can find, because we tend to lump this disease into Chiari 1. I think there's many different types of Chiari out there for sure, because it is a spectrum, as was mentioned before. Thank you.